like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lulin School of Medicine Healthy Longevity webinar. Thank you for being with us. There was a change in our guest. Today we will learn all about glycanaging from Professor Gordan Lauc, and he is the founder and CEO of Genos. But before we start, I would like you to remind you to use the Q&A function to send your comments, which are highly valued, and of course your questions. The short presentation today will be given by Sylvester Wong. He is a research assistant from Associate Professor Dean's Lab, who is a member of CHL, the Center for Health and Longevity, and he recently also served as the head of Department of Anatomy at the National University of Singapore. And his lab is really interested in understanding the molecular and the epigenetic mechanisms that contribute to neurodevelopmental disorders and also cognitive impairment in aging. So, Sylvester, over to you.
Hi, and thank you for the kind introduction. As mentioned, my name is Sylvester Wong, research assistant in Prof Dean's Lab in the Department of Anatomy in NUS, and I'm here to share with you about our lab's research. Our lab is currently researching on dystrophic microglia, and so in this short presentation, I will show you what are microglia and what is the dystrophic phenotype, what it means to the aging field, and what might turn them dystrophic. So let's first look at what are microglia. Microglia are immune cells in the brain that perform many functions. So they monitor their environment with surface receptors and execute immune responses like secreting factors. Microglia are also important in clearing cell debris that remain in the brain after cell death or injury. And more interestingly, microglia are also capable of supporting neurons in development and function. So microglia can behave differently in neurodegenerative contexts. For example, microglia can become active during stroke and may promote a neurotoxic environment. Also, in Alzheimer's disease, chronically stressed microglia may also phagocytose viable neurons. Similarly, in aging brains, microglia also show functional changes. There are more pro-inflammatory responses by microglia and reduction of neuroprotection via neurotrophins like BDNF. And so in our lab, we hypothesize that dystrophic microglia may contribute to age-associated cognitive deficits through BDNF. But first, let's look at what are dystrophic microglia. In this research study, the researchers documented a subpopulation of microglia in the brain that has spheroid formations, twisted and shortened processes, and cytoplasmic fragmentation, and collectively termed these as dystrophic microglia. And these dystrophic microglia with their shortened processes was shown in a different study to also have significantly reduced area covered in the brain and may have reduced neuroprotective capabilities. But how are these cells related to aging? Microglia dystrophy have also been associated with brain aging in the context of humans, non-human primates, and in mice. In mice, Microglia were stained in brain slices, and a large proportion of microglia in the aged mice appeared dystrophic. 73% of microglia were identified to be dystrophic compared to 4% in young adult mice. Dystrophic microglia were also identified in a non-human primate, the marmoset. In the age group, dystrophic microglia were observed in the hippocampus at a higher frequency compared to both adolescent and adult groups. And more recently, a study looked at dystrophic microglia in various regions of the brain in 51 humans. As seen in the middle graph, there is an increase in the percentage of dystrophic microglia in the gray matter of the frontal cortex as one gets older. This relationship was, however, not seen in both the white matter of the frontal cortex nor the hippocampus, suggesting that there could be some regional differences in microglia and its local microenvironment. So why are they turning dystrophic? The literature on dystrophic microglia is very varied and have alluded to several factors that may contribute to the dystrophy of microglia. And one possible factor is iron accumulation. In the study on the left, dystrophic microglia were found to be associated with ferritin immunoreactivity. And ferritin is an iron storage protein whose expression can increase in the presence of high intracellular iron concentration. More recently, the study on the right has shown that iron exposure alone was sufficient to induce a dystrophic-like morphology in microglia cell culture. Iron accumulation in dystrophic microglia is interesting because iron and ferritin expression were found to be increased in the microglia of the aging brain compared to normal adults. And because of these studies, our lab is interested to study 1. How iron contributes to microglial dystrophy. 2. Which factors can inhibit or reverse dystrophy in microglia? And what is the unique molecular profile of dystrophic microglia that we can use to distinguish them from other microglia? And ultimately, how we can help promote healthy brain aging. Thank you all for your kind attention.
Thank you so much, Sylvester, really showing that microglia might be a contributor to cognitive decline. But I think that's to be shown. Now over to glycans. It's my real pleasure to have Professor Gordan Lauc here. He is a professor of biochemistry and molecular biology at the University of Zagreb in Croatia. And he is also the founder and CEO of Genos since 2007. And he is also the director of the National Center of Excellence in Personalized Healthcare and holds an honorary professorship at the King's College in London in Europe since 2015. And in 2020, he joined Glycanage, and he is there the chief scientific officer. And Professor Lauc really published an enormous amount of research articles, more than 200, and has lots of citations, over 10,000. And he is the PI and the co-PI in numerous projects and excellent projects being published. So thank you so much, Gordan, for being here. And please let us know what really glycans are doing with the immune system and if we can overcome inflammageing.
Thank you for the kind introduction. And I will try to explain something about glycans and whether we can actually make a decision to change our glycans, and can this help us to revert glycanaging and stop inflammageing. So we know that lifestyle is the most important element of healthy longevity. But here it's important to know that healthy longevity is not about living forever. It's about staying healthy as long as possible. And 6 years ago, when glycanage was just an early discovered biomarker, we did an interesting study together with the Daily Mail. They sent us samples of 6 individuals, all age, I think, 54 at that moment. And we tested their glycanage, and they all came very different. And all these differences could actually be explained by the lifestyle. And particularly sad part of this story was this lady who was over 20 years older than her chronological age. And she was, of course, very upset and actually angry when she got the result. But sadly, this lady died 2 years ago. So I don't say that everybody with a bad biological age will die quickly, but this is definitely something which is indicating there is something wrong and there is a risk we should be aware of. A lot of studies has been done to show that healthy lifestyle can extend our healthy lifespan. And even in our 40s and 50s, if we decide to change our lifestyle and we have healthy habits, these healthy habits add more and more years to our lifespan, but also to the healthy lifespan. So we are staying healthy longer if we live a healthy life. But living a healthy life is not easy. And I think the best example are these two famous monkeys who are, this guy is on a caloric restriction for, I think, close to 2 decades now. And he definitely looks younger, probably feels younger, but he's hungry for the last 2 decades. So maybe this is not the ultimate solution. And definitely it's not, because each of us is different. There is no standard human. So there is no rule which everybody should follow. We all know that it's diet, physical activity, sleep, stress, which are important. But it's very difficult to take care of all of them. And not all of them are equally important for each of us. There are some people to whom the diet is the dominant factor in biological aging. Some other people, it should be stress or sleep. And we are all different. And we have to find out what is the difference and not only try to be these unhappy, calorically restricted monkeys. But the problem in modern medicine is that we are lacking good biomarkers. We know that up to 80% of all smart drugs are being wasted because they're being given to the people who do not respond. And the same thing is for lifestyle interventions. So many people try to do difficult interventions to which they do not respond. And then they change, and eventually they try to find what actually works. And the scientists have tried really hard to find biomarkers, but we still do not have them. And I think one of the reasons was that too many people focused on genes. We sequenced millions of people, and we have very little translation of this knowledge about genetic polymorphisms into the real life. And one of the key reasons for that is the genes do not define everything. Actually, only about 20% of longevity and 30% of complex diseases is genetics. And what we see on this slide are two pairs of identical twins. Every single letter in their genomes are identical, but they definitely have a very different lifestyle, life trajectories, either because of some epigenetic, bizarre lack of luck, I would say here, or lifestyle choices. And something which is missing is most of the studies where people try to identify differences in people are glycans. And glycans are actually responsible for a large part of inter-individual variation. So the reason why we are all different and not all the same is by large glycans. And everybody actually knows that, but people don't call them that way. For example, blood groups are glycans. A, B, and O blood types are different glycan structures. So I usually try to make an analogy and say that the gene is an information. It's a blueprint. It tells us how this Batmobile should look like. And if you make it only of amino acids, just a polypeptide part of proteins, then we will have a basic car like this Yugo, which was made in 80s in former Yugoslavia. But if we do all the post-translational modifications, if we add all those extras which come after translating genes into amino acids, and the largest part of that is glycans and glycosylation, then we have a fancy hypercar like this Rimac Nevera, which is currently being made in Croatia, the fastest electric hypercar. And then there is epigenetics, which is again information, but it's an instruction booklet which tells us which part of this genetic information to use when and how. So we have proteins coded by genes, and then we have post-translational modifications, which glycans being the most important. And when we look at the molecular level, this rotating figure in the middle, this is the famous S-glycoprotein, which has paralyzed the planet for the last 2 years. And all these hairy things on the surface, these are the glycans. And at the beginning of the pandemic, even the director of NIH was writing about the S-glycoprotein without a single glycan on its structure. And yes, it is possible to study S-glycoprotein without glycans, but this would be very similar to studying this poor bird without a single feather. You can do a lot of good research. You can study this bird walking. You can study the physiology of that bird, but you would never see this bird flying because it has no feathers. The same goes for proteins. This is the real S-glycoprotein. We cannot understand the function of S-glycoprotein without its glycans. And not only S-glycoproteins, the majority of proteins are glycosylated. And the glycans are the ultimate layer of molecular complexity. Unfortunately, in the same time, the most neglected molecules of cellular communication. And it's not only me claiming that. There are a series of documents, both in Europe and the US, stating that glycans are directly involved in the pathophysiology of every major disease. But glycans are difficult to study. They're chemically very complicated. Different site of a protein can be glycosylated. Different glycans can be attached to a given site. And doing it in a high throughput is really demanding. And there are not many labs in the world which are doing high throughput glycomics. And my lab was actually the first to move in that direction over a decade ago. And we already analyzed over 150,000 different people. But not any people, but people from the best characterized genetic, epidemiological, clinical studies all around the world, in collaboration with some of the leading researchers globally. And this enabled us to accumulate a huge amount of information about glycans and how they're relevant for human health. And one of the proteins we focus on are immunoglobulins, antibodies. Antibodies do not function if they're not glycosylated. So glycosylation is essential for antibody function, and different glycans can be attached to this conserved glycosylation site at Fc domains. So they're not so much important for the recognition of the antigen, but they're practically regulating all the effector functions. So what will happen after the antibody binds to an antigen? And they have multiple roles in balancing information. We published a very comprehensive review last year in Comprehensive Glycoscience. It's freely available on our website, so if you're interested, you can download it. And inflammation, which is regulated by IgGlycosylation, is one of the main drivers of aging. There was a cover story of the New Scientist approximately 2 years ago, which said you're only as young as your immune system. An important part of this youth of the immune system is glycosylation. And this glycosylation is extremely complicated in the way how it is regulated. By doing a series of so-called GWAS studies, genome-wide association studies, we mapped a network of over 40 genes which regulate IgG glycosylation. And some of these genes are known risk factors for different diseases, including some expected ones like type 1 diabetes, lupus, Crohn's disease, and so on, where we know antibodies are important, but also some unexpected like a Parkinson or dementia or the cholesterol level, which we have no ideas how our glycans actually, how our antibody glycans associate with them. But we know there are at least 94 different phenotypes for which there is pleiotropy between genes which regulate these 94 different phenotypes, also regulate IgG glycosylation. And it's not only polymorphisms in the genes, it's also epigenetic regulation of all these genes, and also direct environmental factors which affect glycosylation. And these three things, genetics, epigenetics, environment, they integrate into a chemical structure, which are molecular effectors that modulate function of proteins, in this case, immunoglobulin G. And we should be aware that alternative glycosylation, meaning adding a different glycan to a specific glycosylation site, is functionally analogous to coding mutations. We know that a single coding mutation can kill a person because you have a wrong amino acid, you have a non-functional protein, you can be dead because of that. And alternative glycosylation can have exactly the same functional consequences. It can make protein function in a different way. But this is not inherited in the Mendelian way, meaning a mutation changes a protein, and then this transfers from a parent to children, but it is inherited as a complex trait, as a multiple genetic loci, their polymorphisms, epigenetic regulation, integrating to make a decision which glycan will be added to a specific glycosylation site. And for example, age is a very important factor here. When we started to look at the large cohorts, we realized that people really change with age. And initially, this was a burden to us because we had to carefully match all the cases and controls in our studies. But then we realized that there is actually an important information in glycans related to aging, because the pace of aging was not only determined by the chronological age, but also by the lifestyle. So this would be correlation between a glycanage and a chronological age. And this dispersion is decided by individual choices, by the lifestyle choices, how people live. And when we published this, editorial was published for our paper saying that, actually asking, are glycans the holy grail of biomarkers of aging? And now, nearly 10 years ago, I would say that they were probably right. The glycans are extremely important biomarkers of aging. We translated this into a commercial glycanage test. And of course, I have to disclose that I'm an inventor of the test. I'm an inventor on the patent. I'm co-founder of Glycanage. I'm heavily conflicted here. But I will try to present only the hard science and give the references for everything that I'm saying, so you can make your own informed conclusions. So the glycanage clock of aging is based on individual glycan structures. We measured 24 of them on immunoglobulin G. And we know that this type of structures, which are known to suppress inflammation, are dominant in young people. And these type of structures, which promote inflammation, are dominant in older people. So we consider this biological aging the change of these young into old glycans, for which we know they have a functional consequences. And we published many papers on the topic, over 200 now, and we really tried to publish everything which is important in a good journal. So we try to publish in Circulation Research, Gastroenterology, a couple of Nature series journals, to have solid evidence that what we are actually showing is the truth. And what we learned is that people with different diseases, and on this slide are lupus, ulcerative colitis, Crohn's disease, rheumatoid arthritis, Hashimoto, type 2 diabetes, colorectal cancer, they all change in the same direction as aging. So the last 4 bars are aging, different glycans, this effect of aging, this effect of different diseases. So healthy people look younger in their glycon than people with different diseases. And we know that these glycans are functional effectors. They actually regulate inflammation. And I don't have time to go through many stories. I will just quickly show you 2 of the stories we have. One is cardiovascular diseases, where we have initially shown that IgG-glycan composition today strongly correlates with the cardiovascular disease risk score. So this is a prediction of future cardiovascular risk. And then we looked into historical samples. There are samples in different biobanks collected 20, 30 years ago with the clinical follow-up. And so we had samples at baseline, and we knew who will have a heart attack or stroke in future. And the predictive power of the IgG-glycan was equally good as the entire AHA score, while for women, just a single glycan structure was even more predictive than the AHA score. And then subsequently, by studying 2,000 twins in 3 time points over 20 years, we actually showed that glycans today predict development of hypertension in the future. And the question is there something which can be done? So if you repair glycans, would you decrease the risk of hypertension? And here we teamed up with Phil Scholl, who did a very interesting study on mice. So he was feeding mice with a precursor of cellulation, the MANAC, which salvaged the IgG cellulation, because normally when you feed mice with a high-fat diet, they become obese, they lose sialic acid on immunoglobulins, their immunoglobulins become old, and they develop hypertension. So when supplemented with the MANAC, mice did become obese, But they did not lose salic acid because it was protected with addition of MANAC, and they did not develop hypertension. So at least in mice, by just improving glycans, you can actually decrease the risk of developing hypertension in the future. Similar thing is for diabetes. Looking at glycans today, we have very good predictive power whether somebody will develop diabetes in the future. And by looking at the same 2,000 twins in 3 time points over 20 years, we have shown that these glycans actually change gradually as somebody is coming close to their diagnosis. So these are people diagnosed with diabetes, these are healthy people, and as time to diagnosis is approaching, they change more and more in the direction of this disease phenotype. So these changes are part of the disease development in diabetes. And also, they're predictive of both prevalent and incident complications. So looking at glycans, we can see whether somebody has or will develop complications. So to put this whole into a single slide, the idea is that we don't have to wait for people to become ill to do something, because normally people go to hospital when they have pain somewhere in the body, they don't function normally, but this is usually beyond the point of no return. So the complex diseases, the chronic diseases, once you get a diagnosis, more or less you keep it for the rest of your life. Just your symptoms get improved. But if we identify people before, and the disease is not 0-1 process, there is a long series of molecular events which lead to a disease. If we find the people here with only biomarkers change, while the molecules which contribute to disease are changed, then by preventive interventions, either lifestyle or the pharmacological interventions, we can change these damage-causing molecules and prevent the disease. So can we do it with glycans? Can we revert the glycanage clock of biological age? In this way, stop inflammation. So as I mentioned, glycanage is now a commercial product. It's available in hundreds of clinics globally. And with some of them, we collaborate with years. And for example, with Joseph Raphael in New York, the majority of his patients would be 20 to 30 years younger than the chronological age. And normally, when somebody comes into the praxis, they will be more or less the close to chronological age. And with time, this can improve for even a couple of decades. And there are many prominent individuals like Professor Tim Spector. Also, we are tracking him for nearly a decade now. He was reasonably well for a long time. Then he got a difficult period in his life, got older, make a decision to change. There is Instagram Live that 2 of us recorded about what he did. And then he improved for 11 years in a half of a year. So things are doable. Now the question is, what works for each of us? One important aspect are hormones, especially in females, because what we see, blue are men here, red are females. While men age more or less linearly with age, women have this strange acceleration of aging around the age of menopause. And estrogen seems to be dominant factor here. We had a very nicely designed placebo-controlled randomized trial of estrogen suppression and supplementation, and it's published. I will not go into details. The main message is that if we lose estrogen, women would age approximately 30%, 9 to 15 years in 6 months. But if supplemented with estrogen, this does not happen. When the estrogen normally returns, everything goes back to normal. So estrogen is both sufficient and required for proper IgG glycosylation. And this is also opening an idea of early diagnosis of the perimenopause, because the perimenopause is a difficult period between the regular cycle and the menopause, where the hormones go crazy, similarly as in the puberty. And the measuring the hormones doesn't really help much because they change so much, and it can even be higher at the given moment. But glycans on IgG are averaging this concentration in a couple of weeks. And then we see a very good prediction of the early symptoms of perimenopause. Actually, sometimes the change in glycans is the first symptom of perimenopause, because we have women aging slowly, then rapid acceleration, and then aging slowly again. And on an individual level, this would look, for example, like this. So women with very good glycanage, extremely quick increase, so maybe up to 2 or 3 decades in a year or half a year, and then this can be improved with proper therapy. So the pace of aging, it's much faster in the perimenopause than before or after. And this was published in iScience earlier this year. Another thing which generally works is weight loss. So obese people, if they lose extra weight, they improve. We have several cohorts of people undergoing bariatric surgery. We had a patient improving for 37 glycan years in 6 months. Of course, bariatric surgery is pretty drastic, but even the regular weight loss helps. So again, the same 2,000 people, 2,000 twins in 3 time points over 15 years. Those who were losing weight, they were aging slower than those who were gaining weight. But there is no magic diet. For example, caloric restriction works more or less in everybody. This was a large study on 700 people on caloric restriction for 8 weeks. But caloric restriction is difficult. Nobody wants to do it. Nobody keeps to it. And actually, usually they regain weight after that. So we did a large study with the 5 different diets, and none of the 5 diets was working for everybody. But each of the 5 diets was working for somebody. So diet is very individual, and people have to find the optimal diet for themselves. The same goes for exercise. Initially, we were thinking that people who are very fit and who exercise regularly would be much younger than people who don't do it. But actually, overtraining is very common. So people often train too much, and physical activity is pro-inflammatory. And if it's not combined with the sufficient relaxation, it can actually drive inflammation and drive glycanaging. So often in people who are overtrained, we see the effect of accelerated aging. Microbiome is extremely important. So glycans and microbes talk to each other on the multiple levels. And we have shown, for example, by a fecal microbiota transplantation, you can change your microbiome, but then this will change your glycome. And this was published 3 years ago in Gastroenterology. And there was an interesting experiment with Tim Spector's son, who was eating only the McDonald's for I think 2 weeks. And what happened, his microbiome got crazy. Because in a normal situation, microbes and glycans on our endothelium are an ecosystem which co-evolves for millions of years. So different microbes feed on our glycans and they feel happy, like this coral reef community. But if you just give a food which is constantly in our guts and which is not actually supporting microbiome, then only the most aggressive bacteria cope, and then they cause also inflammation. So to summarize the story, there is this test which is integrating genetic, epigenetic, and environmental factors into index which can be reliably quantified. It's globally available. You can actually go and buy a set and collect a few drops of blood and mail the samples back. And the key benefit of this is that you actually get feedback, what you are doing. And why I like this Glycanage company is that they actually try to help people by giving them information about what they cannot see in a mirror. And there are sometimes lifestyle changes which change your glycanage. You do something, then you improve it, or you got worse, or you improve it. But it's important to get objective insight into the current state of low-grade chronic inflammation. And there is a quantitative tool which will help you evaluate different lifestyle interventions. So you start something and see whether it works or it does not work. And at the end, I have to acknowledge that our research is very generously funded with a number of research grants. We've got over 15 million euros in the last couple of years, which enabled us to study all those thousands of people and get all this nice information. And I thank you all for your attention. Happy to take questions.
Thank you so much, Gordan. This is a great presentation. Nearly that I'm ordering now a test. But let us go back, because it seems to be such a brilliant biomarker being associated with so many diseases and also predicting the incidence of a disease. Is it a biomarker, or is it also the IgG glycome associated with pathophysiological mechanisms, so really interfering with pathways?
So, I think we have a problem with diseases because we call them traditional names based on somebody who discovered them in a specific situation and then described a set of symptoms. But many diseases have a common underlying molecular pathways, and one of them is inflammation. So I think the reason why glycanage associates, or let's call it IgG-glycan composition associates with the changes with so many diseases is because it really affects inflammation. So IgG-glycans regulate inflammation on multiple levels, and they're used to suppress inflammation. For example, if you give young immunoglobulins to a person as a IVIG, intravenous immunoglobulin therapy, it's suppressing inflammation, and it can be used in multiple different diseases. So ideally, one day we will have a pill which will change a specific pathway, and then it will improve glycosylation of our own immunoglobulins, and we will not have to take hundreds of grams of immunoglobulins from other people. So I think IgG-glycans are functional effectors. They actually affect many pathways.
That makes sense. So, but tell me, how if I would lose weight, lifestyle intervention, etc., you showed that the IgG-glycan is really changing, or there is a change because the biological age is then also lower. How is that happening if the glycosylation is really determined also by, you said 40 genes, etc. You found how is that working? Because through lifestyle interventions, we will not change our genome.
Yeah, but remember, these genes are involved, but each of these genes is coding for a protein which is working in a specific pathway, which is also affected by epigenetics. So it can be a genetic signature which will decrease a specific gene, not only the polymorphism, but also the environment. For example, simple glucose concentration in the long term will have an important effect on glycosylation. So we cannot change our genes, but we can change the way they are regulated, and we can change all these other factors which go into these enzymatic or signaling reactions. So we did heritability estimates for the IgG glycosylation, and different glycans are between 30 and 70% heritable, different component of the glycan, and the glycanage itself is around 40% heritable. So yes, part of it is genetic. You can't change it. It's kind of hardwired, but still the larger part you can affect by lifestyle intervention. But it is not easy, and it's not the same in everybody. So there are some skinny people who have a bad glycanage, and definitely you cannot tell them lose weight because it will not help. So it's very individual. We are still lacking the knowledge to know upfront what is the cause of good or bad glycanage, but we can evaluate effects of interventions. So, for example, I don't know, my glycanage is horrible, and when I learned that, I went hiking as crazy in mountains. I was hiking 3 or 4 hours a day entire summer. I lost over 10 kilos, but I didn't improve much because I was actually over-exercising, and this increased inflammation. So, for example, for me, it's mostly diet. So when I think about what I eat and I am careful, things get better. Some other people, we have people who had, it was just stress. Because stress is such a strong factor in so many different aspects of physiology that people who actually generate this fight or flight response within their own mind, they just think about bad things, and then their organism thinks it's in danger and activates the stress response. It can be cause for accelerated glycanaging. So we are all different.
But you are specifically looking at the IgG form of the glycone. So an IgG belongs to the immune system. So have you also studied, for example, how people react to vaccination, or now with COVID, if it's predictive for outcome of infectious diseases?
So we look also on other proteins. So I was talking about IgG today. So for IgG, we know, for example, in COVID, it very rapidly reacts, especially if people have severe COVID. So severe COVID could age a person for 10 to 15 years in a couple of weeks. When I had COVID, I aged for 5 years, and it's kept for nearly a year. So sometimes there are long-term consequences of infectious diseases. But it's not only COVID. For example, we see it in acute influenza, the viral influenza. But we don't see it in bacterial influenza or in bacterial pneumonia. So yes, acute infection can cause big changes. And some other diseases can cause changes. Some other proteins also change. So it's its entire spectrum of changes can be caught in glycans, not only IgG-glycans. Now I'm talking about IgG-glycans today, but for example, one very interesting protein is alpha-1 acid glycoprotein, which is one of the acute phase proteins, which also changes glycosylation rapidly when something is going on. But for that protein, we don't really know what glycans do. For immunoglobulins, we know that glycans form, they affect conformation of the Fc domain, which then decides to which receptor will antibodies bind. Because, antibody does not know what to do when it sees an antigen. An antigen can be bacteria, can be food, can be a virus. And the response should be very different. And this knowledge is added to antibodies by glycosylation.
Okay. So the color of the feathers will determine if the IgGs then react to certain antigens. Okay.
It's for example, there was a very interesting paper recently showing, it was not ours, that after vaccination, you develop antibodies with different glycans than after COVID. And this could be one of the reasons why vaccination doesn't really hold for very long. Because, all these mRNA and vector vaccines, they're actually producing antigen in the wrong places in our body. And our immune system is confused. So why is now my heart producing a strange protein? I don't want to attack my heart. And then it's actually suppressing antibodies by putting so-called calming down glycans, putting glycans which are not promoting inflammation. And then you see a virus, and then you're completely confused because you have antibodies which are not supposed to kill the target. And then you see we know so little actually about the immune system.
So in the glycan age, because you're building a clock, or you have a clock, an aging clock, and I would say the not entire community is talking about clocks, but have you looked at within one individual, if the glycan age is high, if then also other clocks, for example, the epigenetic clock is high or a metabolomic clock is high? Have you looked at that?
So generally they correlate.
Okay.
No, they all correlate with age. So they will also correlate to each other. But on an individual level, they can be different. So we have seen people with a relatively good epigenetic. But of course, we have to be careful with the epigenetic clock because there are many different epigenetic clocks. Yeah. So for some of the clocks, they could be bad, and then the glycans could be good, or the vice versa. So I'm not saying that this is the universal clock of biological age, because we don't know what is biological age. There's no golden standard, so we cannot say which of the clocks is better. And I'm not too happy with this word clock, because what we are trying to say with glycans is, we know that these old glycans, they promote inflammation. The young glycans, they suppress inflammation. So in some situations, we want to promote inflammation. So if there is a acute infection, we need to promote inflammation to kill the bugs. But if the low-grade chronic inflammation is caused by obesity or by stress or the wrong microbiome, then we would like to suppress this inflammation, because inflammation is doing damage, especially the low-grade chronic inflammation. So it's I'm not trying to compete which clock is better. I'm just saying the glycans have an important set of information. Glycans, immunoglobulins, have an important set of information about the we can call it age of your immune system or pro-inflammatory direction of your immune system. And, it can help to know that information.
Yeah. Yeah. My last question before handing over to Shamila Kumar to really know what the audience would like to know is, look, I'm a clinician, and I think there are lots of clinicians also in the audience, and you showed data where the glycanage or certain measurements really predicted with an AOC the incidence of diabetes and cardiovascular disease was 0.8. This is fantastic. Why hasn't been what's needed that it's been implemented in clinical practice? Because I would say none of cardiologists would know if I would just call them, do you know what it is? They wouldn't know. So what is needed?
So this is not an easy task, because to have something validated as a diagnostic marker, you have to have all the reagents and instruments. In Europe, it's CE validated for IVD, and glycans are not being analyzed in routine laboratories using routine instruments. So we started the Human Glycon Project, which is integrating efforts of people working in the glyco field to work together with the instrument providers to develop instruments which would be IVD validated, to develop all the reagents, and then we will be able to use the data we have. But then again, we will have to redo it the way to validate the clinical biomarkers. So this will take years, probably over a decade. This is why we have registered the glycanage as an aging test, because this is not a diagnostic test. But within the glycanage, you get the total profile of IgGlycans. So glycanage as a company cannot make any claim, medical claim, but any clinician can look at the data and use it as any other non-diagnostic test to see whether there is some beneficial information for a patient.
Yeah, thank you. Very clear. Shamila, over to you. Thank you for joining.
Thank you, Professor Maya. Thank you, Prof Lauc, for your wonderful presentation. There's an overwhelming response from the audience. Let me just start with the very basic question they've been asking. In your opinion, what makes N-glycans a reliable biomarker for aging?
Well, they strongly correlate with age, but methylation, the original Steve Horvath's clock, is actually better in predicting chronological age. The prediction for chronological age is better, but glycans are actually better in predicting, in explaining the difference between chronological and biological age. Because, I don't need a test to tell me my chronological age. I have it written in my ID card, and we all know our chronological age. The only practical use of chronological age is, for example, when you have illegal immigrant and you want to show whether that person is a minor or an adult. And some countries use methylation age for that. On the other hand, glycans are giving you information about this aberration from your chronological age, which I think is valuable. Why do I think it's good? Because we studied thousands of people and we published many papers showing very strong and consistent correlations. So I think it's good. It's not ideal. It's never 0-1. There will be people with the good glycans who will develop different diseases, or people with these old glycans who will live longer. But there's a lot of information potential there.
And then there's another question saying that is glycosylation a cause or a consequence of aging?
I would say both, because by being pro-inflammatory, they promote inflammageing, they promote inflammation. But then again, there are other aspects of aging biology which affect IgG glycosylation. So they're in part consequence. For example, obesity. Usually people, as they get older, they become a little bit fatter, and this accumulated, actually the old fat, which has been there for years, is sending pro-inflammatory signals, which is then changing the IgG-glycan. So I would say both cause and consequence.
There are also concerned that what do you consider a good amount of physical activity that can reduce inflammaging?
So I'm not an expert in physical activity. When I talk to people who are good in that, they say it has to be individually analyzed for every given person. And it's somebody who is very sessile, if they start exercising every day, they will do more damage. And so it has to be measured on every individual. It has to be individually determined, and then to see what. So maybe on average, what they claim would be maybe 2 days of strength training and maybe 3 days of slight cardio, even hiking would be good. But it has to be individually tuned. And of course, it's very different than somebody who's in their 20s or 30s or 60s or 70s. So.
But may I ask on this, if we don't know what we should do in terms of exercise, looking at glycanage, for example, when should you remeasure the glycanage when you are then exercising? And is that different if you are doing resistance exercise training or cardiac training?
So I would say 3 to 4 months. But in some cases, we have seen effects earlier. So the IgG has a half-life of approximately 3 weeks. So it does take time for it to change, and the exercise takes time to change condition in a body. So it's very hard to see effects before 2 months. In 3 to 4 months, there should be visible effect if it's good condition. So based on our limited experience, this high-intensity interval training is mostly beneficial for most people, and it gives quick response.
There's another question also. How does chronic inflammation contribute to the aging process in humans in general?
So inflammation is a kind of reset. So if there is something, if there is an injury, if there is an invasion by pathogens, you send in your components of the immune system, white blood cells, and so on, and they destroy this part of the tissue. So you come in, see the damage, destroy it, and then the organism rebuilds. The problem is, each time you destroy and rebuild, it's never perfect. It leaves a little bit of a scar. It causes some additional mutations and so on. And then low-grade chronic inflammation, which goes for a long time in the different parts of the body, is kind of continuously adding scar on scar on scar. And it's promoting hypertension, insulin resistance, and everything which leads to complex diseases.
They're also questioning that how does de-glycosylation occur during aging?
So it's not removal of glycans, which are already there. Mm-hmm. It's just, since we are always building new immunoglobulins, when we make the new ones, we make them to be more pro-inflammatory with the shorter glycans. So it's not just the chemical damage like the glycation, which is this happens in diabetes. When you have too much glucose reacts with the proteins, and then you have this advanced glycation end products. Here, it's a decision by the immune system to make the pro-inflammatory antibodies. So it's not the damage, it's not the chemical damage, it's a biological decision to make pro-inflammatory antibodies.
They are also questioning that is the molecular machinery behind a precise glycosylation of protein is well studied?
Well, we know all the enzymes, we know the pathway, we know very little about its regulation. So we don't know how these things are regulated. And when we did all the GWAS studies we did, so the genome-wide association studies, we found that the regulatory mechanisms are not actually in the synthesis pathway. Some of the regulatory genes are not even expressed in the B cell. So it's really a very complex decision made by the immune system how to glycosylate the protein. And this decision is really very complex because, in biology, we, as a very long-living organism, are competing with the pathogens which will make 1 billion offspring in a day. So we cannot compete with the genetic variation. We compete with our immune system. And our immune system is very sophisticated. It knows how to fight antigen. I think, the best example is we can have, influenza and be nearly dead for 2 days, barely alive. And 2 days later, it's like nothing happened. Your immune system fought off the virus. And part of this immune system is glycosylation. And I would repeat, we know very little about how it functions. And I think we have learned this very, very clearly in this pandemic, that we really don't know how our immune system works and when will it work and when it will not work.
They also asking that glycated hemoglobin is well associated with diabetes. But how does this compare with glycated IgG in diabetes?
So these are two completely different things. So glycation is a chemical reaction between glucose, and it is prominent in diabetes because in diabetes we have more glucose than in a normal condition. So glucose, which is an aldehyde, reacts with amino group, and then there is this Amadori reaction, and you get this advanced glycation end products. This is a chemical damage. And we measure it, glycated hemoglobin, to know the average concentration of the glucose in the past period. Glycosylation is a regulated enzymatic process which has nothing to do with the glycation. They just have a similar name, but it's a biologically completely different process.
Very clear. Shamila, last question.
Okay, before I leave, are there other proteins other than IgG whose glycosylation can predict negative outcomes? Or why is it just Ig alone?
No, well, we mostly study IgG because it's the most abundant. It has a simple glycosylation. This is why we started. For example, when you look at the total protein glycom, of all proteins, it's very predictive of diabetes. Alpha-1 acid glycoprotein is also very predictive of diabetes and some inflammatory diseases. Many other proteins are predictive for other things, but we have to study them. We cannot know without studying.
Thank you so much for answering the questions.
Thank you.
Thank you so much, Gordan. It was a very impressive talk. I've never seen so many data published so highly, really showing that a biomarker is so associated not only with age-related diseases, but also with the incidence of age-related diseases. So I hope you as audience, you also enjoyed the talk. I would like to ask you to use the chat function and choosing the panelists and all attendees option. If you have feedback, comments, other questions, please leave them with us, and we will leave a couple of news from our center and our School of Medicine after the end credits. And of course, you're interested who is next in our series. It's in April 14, so next week. And my guest will be Professor Giovanni Grillari. And I will leave you with a final video, as always, and you will see Dr. Charles Godfrey. And I can tell you it's a person with a very big heart. Take care.
Happy birthday to you.
God, I look around this room. I wish I was 3 years younger.
Come on, who wouldn't want to see Dr. Godfrey? He's totally with it. He's even got Trump jokes. Albany Medical is just one of the clinics he works at, and Dr. Godfrey doesn't just aim to make his clinic great again. At 100 years old, he's still making patients feel great again through physiotherapy.
When I started, it was bake and shake.
How so? What do you mean?
You heated the patient and you shook him, or massaged him. Bake and shake.
Gosh.
Now a good deal is more concerned with prescribing and making sure they're doing their exercise properly.
What's your secret?
My secret? Somebody's baking and shaking me. I don't know what my secret is.
Carol Denvers has been his receptionist for 17 years.
What is special about him as a doctor?
His heart. Big heart. big heart, I tell you. He just so compassionate, just sweet, this greatest person that I've actually ever met. Like, I love him.
Ali Chiasson, CBC News, Toronto.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.